Hello students, welcome once again to my YouTube channel, and in succession to the videos what I have been taking up in physiology, in succession to those videos, today's topic of the discussion is one of the very important and a very favorite questions what has been very frequently asked in most of the professional theory exams, that is about the physiology of salivary secretion. So today's topic I'll be mainly dealing up with how the physiology of the salivary secretion is there and how it is being synthesized. So students, starting with, so before getting into the details of as to how the saliva is being secreted, let us now look into that in the oral cavity, our oral cavity is continuously bathed or it is entirely moistened with the thin layer of saliva which keeps the oral cavity moist. So how and from where these salivary secretions are there? So for them, our body, in our oral cavity, there are basically three major salivary glands. So three major paired salivary glands are the parotid, the submandibular and the sublingual salivary glands. The submandibular, it is sometimes also referred to as the submaxillary gland. So these three are the major paired salivary glands, and when looking at the secretions, what kind of the secretions they predominate with? Predominantly the type of secretions what they secrete, it is the parotid gland which has the secretions which is entirely rich with water and electrolytes. So the secretion is particularly called as the serous secretion, which is rich in certain enzymes, digestive enzymes are there, and it is rich in water and electrolytes. Moving on to the sublingual salivary gland. The sublingual salivary gland, it is the secretion of it is mostly a kind of a sticky or a mucinous substance which is called as the mucin is being secreted. So the secretion of the sublingual salivary gland, it is having a mucus secretion. So it is the secretion of the sublingual salivary gland, it is the mucus secretion. Whereas when looking at the submandibular salivary gland, so in the submandibular salivary gland, since the parotid was predominantly serous, the sublingual was predominantly mucus, so the submandibular salivary gland is having somewhat of a mixed secretion. That is, it is none of the other, it is neither having a predominantly serous or nor a mucus, it is somewhat having a mixed secretion of it. Now, out of when looking at the entire bulk of the saliva, so the entire bulk, the 75% of the saliva, what is being secreted in the oral cavity, it is from the submandibular salivary gland. So these are the submandibular salivary glands which constitute to about the 75% of the salivary secretion, and the remaining, that is the 20-25%, it is from the parotid salivary gland. The 25% of the salivary secretion is from the parotid, the 70% from the submandibular, and the rest remaining it is from the sublingual salivary glands. In addition to them, there are certain minor salivary glands which are located in the oral cavity, particularly in the oral mucosa, the pharyngeal outlets. So there are certain minor salivary glands, and the best example for it is the glands of von Ebner, which are located in the posterior part of the tongue. So these are this was a brief overview about the salivary glands. One of the very important questions what is very frequently asked in most of the competitive exams is about the ducts. Since salivary secretions, the salivary glands, so salivary glands, they are the exocrine glands. So exocrine glands are those glands, as you all might be very well aware, that exocrine glands are those glands which are getting their secretion poured through a duct. So they are also called as the duct glands because they secrete their secretion by the means of a duct. So for their secretion, there has to be presence of a duct which is going to pour the secretion in that particular area. Whereas on the other hand, there are certain endocrine glands which directly release their secretion into the bloodstream without the need of the duct. So the endocrine glands, they have also been called as the ductless glands. So saliva, since it is in salivary glands, they are exocrine, they are having a particular duct for getting their secretion discharged. So similarly, now these parotid, submandibular and sublingual glands, they are having their specific ducts, and these ducts, they have been very frequently asked in the various competitive exams. So students do remember that is the parotid gland, the duct what through which the parotid gland is going to drain its secretion into the oral cavity. It is the Stenson's duct. Now this Stenson's duct, it drains its secretions opposite to the second maxillary molar. So the second maxillary molar is the site of the opening of the Stenson's duct through which the parotid releases its secretion. Moving on to the sublingual salivary gland. The sublingual salivary gland, the duct through which it releases its secretion, it is the duct of Riveneus, and there are approximately these duct of Riveneus, they are about mostly 10 in number. And moving on to the submandibular salivary gland, the duct through which it releases its secretion, it is the Wharton's duct. So these three ducts you must remember: the parotid, the Stenson's duct, which opens opposite to the second maxillary molar. The opening of this Stenson's duct is opposite the second maxillary molar on the buccal mucosa near to it. The sublingual, it is the duct of Riveneus, and the submandibular, it is the Wharton's duct. Now moving on to the histology. Histology, whenever we look into histology, we go through the cut section, how a particular organ or a tissue appears when viewed under a microscope. So when viewed under a microscope, what do we find is that how the cells are appearing, as to how the cellular arrangement of the cells and the tissues are there. So when looking at the histology of the salivary gland, the basic unit of the salivary gland, it is called as the salivon. Just as the basic unit everywhere, for a kidney it is a nephron, for the nervous system it is the neuron. Similarly, in case of a salivary gland, it is the salivon. Now this salivon is going to have three components. These three components can be summarized as in the form of: first is the acinus. The salivon is made up of an acinus, it is made up of a striated duct, it is made up of an intercalated duct. Now, the secretion through which the acinus is releasing its secretion, it is called as the primary secretion. And actually, what is this acinus? So acinus, it is some kind of a sac-like structure, and this sac-like structure, as you can see in this diagram what I have drawn, the blue colored, this circular thing, it is a sac-like structure which is lined by pyramidal and myoepithelial cells, which are going to release predominantly the serous type of secretion, that is predominantly watery secretion along with electrolytes and digestive enzymes. So acinus, it is a sac-like structure, and the secretion through an acinus, it is always called as a primary secretion. Whereas there are, in addition to it, there are certain ducts which are called as the intercalated ducts. So intercalated ducts are those ducts which are going to join, which are going to interconnect two acini. So there are intercalated ducts, and apart from them, there is a striated duct through which the entire salivary secretion is being poured. So these were about the type, the salivon, the basic unit of the salivary gland. It is called as the salivon, and salivon comprises of three things. Firstly, it is the acinus, the striated duct, and the intercalated duct. Now, the serous secretion. So serous secretion, when looking at the detailed structure, the gross, in microscopic view, when we find that the serous salivary, the serous acini, they are containing good quantity. They are the most important, the cellular organelle. It is the endoplasmic reticulum, particularly the rough endoplasmic reticulum and the zymogen granules. So these zymogen granules, they contain the digestive enzymes, that is the salivary amylase, through which they discharge into the main primary secretion. So these enzymes, what are contained, it is in the zymogen granules, and the rough endoplasmic reticulums are very abundantly seen in the serous type of the acini, whereas in the mucus type of acini, they are containing the mucin droplets are present, and they are the mucinous secretions are there from the mucus acini. There are certain acini which are called as the mixed acini, because they are having both the elements, both the components are present, that is the seromucus, the serous part as well as the mucus part. So therefore they have been given the name of the seromucus acini. Now moving on to the innervation of the salivary gland. So students, the salivary gland is being supplied by both the types of the autonomic nervous system, that is the parasympathetic and the sympathetic nervous system. So whenever there is an orexigenic stimulus. Orexigenic stimulus is that kind of a stimulus which is going to cause increase in an appetite or causes the increase in salivary secretion. So whenever there is an orexigenic stimulus, for example, the sight, the smell, the taste, and the chewing of the food, that causes the afferent impulses to reach to the higher centers of the brain to cause the excitation through which the impulses will carry, and finally they will reach to the respective glands to cause that secretion. So the innervation, so here we can, as I have drawn the schematic diagram, you can see, so this is the schematic diagram of a brainstem, and the important parts of the brainstem, that is the midbrain, and the pons and medulla are there, and within it there are going to lie the two very important nuclei, that is the superior salivary nuclei and the inferior salivary nuclei. So since there are three major salivary glands, that is the parotid, submandibular, and the sublingual salivary glands, so what is going to happen is the taste sensations from the posterior 1/3 of the tongue, they are going to take the impulses through the glossopharyngeal nerve to the higher centers in the inferior salivatory nucleus, whereas the taste sensations from the anterior 2/3 of the tongue, they carry the taste sensations through the facial nerve to the superior salivatory nucleus, and in a way they cause the secretion, salivary secretion, and it is the salivary gland, it is the only gland through which the secretion is elicited by the taste sensation or the nerves stimulus is being sent, neural stimulus is being sent to it. So students, very, very important, it is the parasympathetic supply, in the innervation. So what is going to happen is, from the inferior salivatory nucleus, the preganglionic fibers, they are going to originate, they pass through the glossopharyngeal nerve, its tympanic branch and tympanic plexus, and through the lesser petrosal nerve, they finally go and relay in the otic ganglion. Now, from here, the postganglionic parasympathetic fibers, which are shorter, they go and finally reach the parotid gland to cause the secretions from the parotid gland. So this was about how the secretion was carried out in the case of a parotid gland. Now moving on to the next, that is from the superior salivatory nucleus, the preganglionic fibers. So the preganglionic fibers, I have drawn in both the cases with the help of a thick line, whereas the postganglionic fibers I have drawn in the form of dotted lines. So from the superior salivatory nucleus, the preganglionic fibers, they are going to pass through the facial nerve, its branch, that is the chorda tympani nerve, to relay in the submandibular ganglion, and from here, after relaying in the submandibular ganglion, the postganglionic parasympathetic fibers, they originate to finally supply to the sublingual and the submandibular salivary gland. So this was about the parasympathetic secretion, how the parasympathetic nerves excitation, nerve stimulus is causing the salivary secretion. Now, when talking of the sympathetic supply, it is mostly from the thoracic, that is the T2 and T4 segments of the thoracic region. The nerve supply is originating, the preganglionic fibers, they originate and they go and relay in the superior cervical ganglion, and from there, hello Stephen, and from there the postganglionic fibers, they will originate and they finally go and cause the they terminate in these respective glands. So this was about the sympathetic and the parasympathetic nerve supply of the salivary gland. So this is all what I have discussed, and when looking at the normal salivary secretion in an adult human, it is around about 1 to 1.5 liters per day, and when looking at the rate of the salivary secretion, it is about 50 milliliter per minute per 100 gram of the salivary gland tissue. So students, this was one thing, And when looking at the composition of the saliva, so saliva is basically going to comprise the water, which is comprising the 99.5%, whereas in case of solids it comprises of 0.5%, which include both the components, that is the inorganic and the organic. In inorganic components we have the potassium, sodium, chloride, bicarbonate, whereas in case of organic we have the digestive enzymes such as the ptyalin, the lingual lipase, the callicarin, the mucin, the lysozyme. So these are all the enzymes what comprises of the organic component. Now students, the fact what I need to emphasize over here as to what is the primary secretion and the secondary salivary secretion. So students, do remember this fact. Now, the initial secretion, what is there from the acinas, from the acini of the gland, the initial secretion what is there from a particular acinus, it is somewhat having the tonicity which is similar to the blood plasma. So do remember the initial salivary secretion what is there, it is always and always isotonic to the blood plasma. But after secretion, when they reach through the striated duct, so striated duct, what is going to happen is within the striated duct, there is going to be certain influx and efflux of the ions which are going to cause changes in the constituent of the saliva. So the final salivary secretion, what is there after the secretion passes through the striated duct, it is called as the secondary salivary secretion. Now this secondary salivary secretion, which is also called as the final salivary secretion, it is always hypotonic to blood plasma. So students, very, very important, in various PG entrance exams you are questioned that the primary salivary secretion, it is isotonic to blood plasma, whereas the secondary salivary secretion, it is hypotonic to blood plasma. Do remember. Now, what is the main fact what I need to emphasize, that it is the striated duct which is ultimately going to modify the primary secretion, and between the primary and the secondary secretion, the secretions what is there in the later in the striated duct, that is not going to alter the volume of the saliva. It is only going to change the ionic components, or the final composition of the saliva will change in the final secretion. So whatever the volume is being secreted by the acini during the primary secretion, the volume is going to remain the same. But the only modification what is occurring through the ionic exchange, it is the ionic exchange alters, and the volume in both either the primary and the secondary is going to remain the same. So this was the fact what you need to remember. Primary secretion is always isotonic to blood plasma, secondary secretion is always, or the final secretion is always hypotonic to blood plasma. So this is the schematic diagram what I have drawn. This is this particular structure of an acinus, and this acinus is going to release, initially there will be release of sodium, potassium, chloride, bicarbonates and enzymes that is going to release within the lumen of the acinus, and finally when it is going to pass through the ductal epithelial cells, there is an efflux of the sodium and chloride ions through the sodium chloride channels. So there the sodium and chloride, they are being reabsorbed into through the epithelium, whereas from the extracellular fluid or the interstitial fluid, potassium and bicarbonates, they are being pumped into within the lumen. So the final salivary secretion is basically very much rich in potassium and bicarbonate. So final salivary secretion, it is very rich in potassium and bicarbonate, and this is the mechanism as to how the secondary secretion is going to take place by the exchange of certain ions. So students, looking in a brief, in short, I'll be taking up the functions of the saliva. So first function, it is the protection. How protection? Because it keeps the oral cavity moist and wet. It lubricates the oral cavity. It causes the lubrication of the bolus of the saliva is going to lubricate the bolus, and it causes easy passage of the bolus from the mouth to the oral cavity. Moving on to the buffering capacity. Now what is going to happen is that the more the salivary secretion is there, more is the clearance of the food substance or the sugars is there. More the quantity of the salivary secretion, more is the clearance of the carbohydrates and the food from the oral cavity, and hence a greater buffering effect. So another function, it is the buffering effect. In buffering effect, it is that it buffers the, it neutralizes the acidic pH, because the food what is going to remain in the oral cavity, if that food is not going to be cleared off with quick salivary secretion, that is going to cause an acidic content, increase in the acidic content in the oral environment. Now, since the acidic content will increase if the food is not cleared quickly, so if the salivary secretion is increased, if the salivary secretion is more, so more clearing of the food is going to take place, and in a way it can cause the buffering action by the removal of the acidic pH from the oral cavity. So next is the pellicle formation. Now the saliva what is formed, it also helps in protecting our teeth. So the teeth what are present, they are being continuously, they are being lined by they have been coated with the saliva, and this is the salivary pellicle which covers the teeth. And this pellicle prevents the teeth from tooth wear, because the teeth are continuously in contact with each other during sleeping and everything. So that the pellicle formation, the pellicle which bathes the teeth, it is going to prevent the tooth from wear. And whereas there are certain chemicals what are known as tetherins and proline rich proteins which causes the calcium ions, so which causes the efflux of the calcium ions, so they help in the remineralization of the tooth. So finally, it is in the maintenance of tooth integrity. In a way, when the flushing action, quick flushing action is there of the oral clearance of the food is there, so flushing action is there, and since the flushing action is there, so it is going to cause the maintenance of the tooth integrity. Whereas their antimicrobial function of the saliva, it can be looked upon as that the saliva contains a very important compound which is called as the lactoferrin, which binds with the iron, which has a strong affinity towards the iron. Since the bacterial cell wall is containing iron, so this lactoferrin is going to engage those iron from the bacterial cell wall, attract that iron from the bacterial cell wall, and ultimately that will lead to a bacterial cell which will be devoid of a cell wall. And ultimately, in a way, this lactoferrin is causing the antimicrobial effect by degrading the oral microorganisms or the oral microflora, harmful bacteria, particularly the Streptococcus mutans and all. Similarly, there are certain important chemicals which are known as histatins. So histatins, these are the chemicals what are present in saliva, and they are going to inhibit the Candida albicans and the Streptococcus mutans proliferation. So students, these were some functions such as the protection, the buffering action, the pellicle formation, the maintenance of tooth integrity, the antimicrobial action, and finally the taste. Because saliva, it is going to provide a liquid medium to the food. So the food what we ingest, that is being diluted, that is being dissolved in the saliva, and through this dissolution that enters into the taste buds, particularly the folate and the circumvallate papillae. The dissolved food in the saliva is going to reach to the taste buds, and it causes a sensation, it provides the sensation of taste to us. So these were the few functions what are related to saliva. So students, this was a short discussion. I have tried my level best to simplify in a nutshell as to what are the salient points, what you need to pen down during the professional exams if you're asked to write about the physiology or the important the note on saliva secretion. So students, if you do have any queries or comments, you are most welcome to comment me in the comment section. And students who are new to my channel, and if they haven't subscribed yet, do subscribe my channel, and don't forget to press the bell icon so that you can be further updated for my upcoming videos. Thank you for watching.